Please work. Please. This machine modeled parts of the reproductive system before, during, and after conception. We start off with the egg being released from the ovary into the fallopian tube during ovulation. This egg sits at the top of the fallopian tube until it is met by a sperm. After the male erection and ejaculation, sperm travels up the fallopian tube, meets the egg, and becomes a fertilized embryo. This embryo travels into the uterus where it embeds itself in the lining of the uterus. The embryo then begins to divide rapidly, growing and developing into the clumps of cells that would be birthed as a newborn baby. But how does the science really work? Let's take a deeper look.
The first aspect of this machine is the fallopian tube. A ball, representative of an egg, travels through the fallopian tube, or in this project, a piece of PVC pipe. Once in the uterus, a sperm will fertilize the egg, and the cycle will continue. This portion of the machine was directly related to physics. Gravitational force was exerted on the ball, causing it to slide down the tube. The friction force of the tube was not great enough to prevent the ball from sliding, and as such, the ball continued to accelerate until it collided with the wooden ramp. The wooden ramp absorbed the energy of the ball and exerted an equal force on the ball, causing it to travel in the opposite direction in accordance with Newton's third law of motion.
As for my part, I had to make something that would represent the male erection. I decided to make a chemical reaction composed with vinegar and baking soda, aka sodium bicarbonate. Those chemicals reacted in two different reactions. The first reaction is an acid-base reaction, which the vinegar is acidic and the sodium bicarbonate is basic. When these chemicals are mixed together, hydrogen ions in the vinegar react with the sodium and bicarbonate ions in the baking soda. The products made in the initial reaction are carbonic acid and sodium acetate. The second reaction is a decomposition oxide reaction. The product of the initial reaction, carbonate acid, immediately begins to decompose into water and carbon dioxide gas. Since the baking soda was already in the balloon, I just poured some vinegar into the balloon, which then inflated because of the creation of carbon dioxide. When the balloon had took enough volume, it pushed down a medium marble down the ramp.
I was responsible for the third piece of the project, male ejaculation and sperm traveling to make contact with the egg at the top of the fallopian tube. The sperm is initially deposited high up in the vagina near the cervix and begins to travel up the fallopian tube. Only 1 in 14 million sperm reach the fallopian tube, but once it does, chemical signals from the egg will help the sperm reach it and achieve conception. To model this system, I used physics, with a marble rolling down a ramp. The ramp represented the sperm's path to the egg, and at the base of the ramp, it comes in contact with the egg, representing conception. The ball began in rest and then was pushed forwards by the reaction in the balloon. I used the video to create this distance over time graph for the system. This graph shows a curve, showing that the ball is accelerating as it is going down the ramp, getting faster. Since the ramp is curved, not at a constant angle, it can be assumed that acceleration is not constant. The slopes of the curve at the different points show the acceleration, and it is clear that the velocity is increasing as the ramp becomes steeper.
Before I discuss my part of the project, first allow me to explain mitosis as simply as possible. How does this biological process work? Well, interphase occurs before it, leading into 4 phases: prophase, metaphase, anaphase, and telophase. After mitosis, cytokinesis occurs. In interphase, the cell grows, duplicates its DNA, and produces proteins and enzymes, and mitosis soon begins. Prophase is where many structures in the nucleus disappear, including the nuclear envelope and the nucleoli. After being duplicated, there are now 2 centrosomes, which move to opposite ends of the cell, forming pores. Mitotic spindles are released from the pores, and the chromosomes are able to divide into equal sister chromatids connected by a centromere. As you can see in this picture, the chromatids are then lined up at the center of the cell in metaphase. In anaphase, they split. And in telophase, the spindles soon disappear, the nuclear membrane reappears, in addition to the 2 new cells sort of reverting back to how they generally were in interphase, if we're using simple language here. The cytoplasm is then pinched into two separate bodies, and the cell is finally split into two during cytokinesis due to the contractile ring creating a cleavage furrow. The two daughter cells are smaller than the parent cell, but in interphase they will soon grow to repeat this process again. As you could see, my part of the machine was a line of blocks of increasing size falling over like dominoes, although it was not very realistic as there were not as many blocks as cells in the human body. In addition to not looking like mitosis, realistically, it still was able to symbolize the biological process of the zygote, aka the fertilized egg, undergoing cell division and growing larger overall. Here, mitosis can create a human with an identical set of chromosomes in each cell. And now, we move on to our next step.
In order to figure out the velocity of the basketball, the first step is to draw a diagram of what's happening. In this diagram, the ball shown at the top is the initial velocity, and the ball shown at the bottom is the final velocity. Ux is the initial velocity in the x direction, or horizontal, and Uy is the initial velocity in the y direction, or the vertical. Same with Vx and Vy. They are the final velocity in the horizontal and the final velocity in the vertical. Therefore, the initial horizontal velocity equals the initial velocity cos theta, and the initial vertical velocity is the initial velocity sine theta. The next step is to figure out the givens. You already know the displacement in both the x and the y directions by simply measuring. You know the initial velocity in the y direction because the ball was sitting on a table, so therefore it's 0. You don't know either of the final velocities. You know the initial acceleration in the x direction, which is 0, and the acceleration in the y direction, which is 9.8 meters per second squared, as that is the acceleration due to gravity on Earth. You have time by simply timing how long it takes from the ball leaving the table to hit the floor. The next step is to solve for what you're not given. You can solve for both velocities and the initial acceleration in the x direction by using Suvat equations. The final step is to combine the final velocity in the x direction and the final velocity in the y direction to get the final velocity overall. And by doing this, you can see that the final velocity of the basketball is traveling is at 13.7 meters per second.
The final section of our system is the delivery of the child. In our system, this is represented by a basketball being pushed off the table. In this, the basketball represents the child, and the act of the basketball falling off the table represents the newborn leaving the vagina. The act of pushing out the baby happens when the cervix is fully dilated. When this occurs, this is known as the transition phase. A cervix is a narrow passage that forms at the end of the uterus. Once the cervix is fully dilated, the baby will move down the birth canal towards the entrance of the vagina. In a safe birth, the head of the child exits the vagina first. The rest of the baby is normally completely out of the vagina the next 2 to 3 contractions. The delivery of a child takes an average of 30 minutes to an hour once the final stage occurs.